So hello, I'm Jennifer, and I'm going to share today a project that I was working on in the lab of Robbie Loeth at the University of Geneva. And that is this architecture of a near-native stretch-sensitive membrane microdomain. So all cells must respond to mechanical and osmotic forces in order to protect the integrity of their membranes. They've all needed to develop mechanosensitive mechanisms to sense and respond to these forces in order to survive. So today I'm going to talk to you about an interesting example of a mechanosensor, that is the yeast isosome. So isosomes are stable plasma membrane invaginations found in most fungal species. And in Cerevisiae, they're around 300 nanometers in length and 50 nanometers deep. And they're randomly distributed across the cell surface. So there are several protein families that either stably or conditionally localize to the isosomes. And there are two pyrologous bar domain proteins, PIL1 and LSP1, that form a complex that scaffolds the invaginations. So bar domains are an interesting family of proteins that's known for its role in membrane modifying processes. They function to either sense or drive membrane curvature through their binding and oligomerization. So from the crystal structure of LSP1, you can see the characteristic banana shape of a bar domain protein. And like many other bar domain proteins, PIL1 and LSP1 have this interesting tubulating behavior in vitro, in which they form a patterned array across the surface of membranes. So the curious thing about isosomes is that they scaffold one of the better characterized examples of a membrane microdomain. There are several lipid species that are proposed to be enriched in the isosomes. They're enriched in ergosterol, which is the predominant sterile species in fungi. Also, the assembly of isosomes is dependent on PIP2, both in vivo and in vitro, and they play an important role in sphingolipid biosynthesis signaling. However, the interpretations of all of these data are a bit complicated because of an important problem in membrane biology, and that's the limitation of using fluorescent lipid probes. So if you tag either the head group or the tail group of a lipid, it alters its chemistry and its behavior in membranes. And so there's a lot of controversy in the interpretation of data from these probes. Meanwhile, the disruption of lipid biosynthesis pathways with either drugs or mutants can have wide-reaching off-target effects. What is clear, though, is that functionally, the isosomes have a role in sensing stresses on the plasma membrane. Under steady-state conditions, isosomes sequester several proteins, including SLIM1 and NCE102, and these are TORC2 regulators. Upon various types of membrane stress, including hypoosmotic shock, heat shock, and mechanical pressure, isosomes appear to flatten, and they release their sequestered isosomal proteins, enabling their signaling functions. For example, SLIM1 goes to interact with TORC2, which then goes on to initiate processes that impinge on plasma membrane homeostasis. Now, our story begins serendipitously with Robbie's student Ming, who was doing structural studies on TORC2, a multi-protein complex that's one of the main focuses of Robbie's lab. After pulling down TORC2, Ming captured these tubules on his lacy cryo-EM grids, and thinking that they might represent a higher order complex of TORC2, he sent these grids to Heidelberg to collect a cryo-EM dataset. However, it quickly became clear that these were not TORC2 at all, but instead a contaminant. And that contaminant turned out to be the isosome proteins PIL1 and LSP1. This dataset then sat on the shelf for a short while, but it eventually became my main project. So to solve the structures of the isosomes, I went to learn helical reconstruction from Ambroise Defosse in Grenoble. And in the end, we were able to solve 9 different tubule structures that have different diameters and helical symmetries. The diameters range from around 310 to 370 angstroms, which is actually well within the range of the diameters of the isosome furrows observed in situ by tomography by the Kukulski group. Now, within the protein scaffold, we saw something very interesting. We could see this lipid bilayer inside our tubule. So because these filaments were extracted using a relatively low detergent buffer, which was another happy accident, we believe that these isosomes retain the native plasma membrane that was associated with them in vivo. So let me orient you to what we're seeing here. So this is a central slice through the isosome tubule, and you can see the two layers of the two leaflets of the lipid bilayer. I've highlighted in blue the PIL1 LSP1 lattice, which is bound to the cytoplasmic leaflet, highlighted in yellow. And the exoplasmic leaflet, highlighted in pink, is trapped inside the tubule. So here we've basically captured a nearly native plasma membrane microdomain. So what can we learn about it from our structures? So what I've done here is turn the isosome structure on its side, and I've made these 1 pixel slices through it. In the first membrane slice, that we can see that buried within the membrane density, there's a well-resolved density that corresponds to an amphipathic helix. Here's the more traditional view of that. Here I've built a protein model of PIL1. So each unit of the lattice is a dimer of 2 PIL1 proteins. And when we turn our view to the side here, we can see these 2 amphipathic helices. We can also see, if we zoom in, these additional N-terminal residues that come back out of the membrane and sit between PIL dimers to form a part of the contact site with the neighboring PIL dimer. And here's a better view of those contact sites. There are actually 3 sites, this one between the N-termini, which is a bunch of weak interactions between them, and the other 2 sites at the bar domain tips. I also want to show you another very interesting feature of the membrane in these tubules. As we continue through the membrane beneath the amphipathic helices, we actually see this distinct pattern of voids that continues through the cytoplasmic leaflet. But as we get to the exoplasmic leaflet, there's no such pattern. It's actually quite smooth. And here's the zoom of these panels. So this was very exciting because I think it's an entirely unexpected visualization of the effect that a scaffolding protein is having on the lipid behavior. But what's going on here? So one possibility is that we're seeing a signature of sterile molecules that are supposed to be enriched in the isosomes. Sterols have a relatively weaker electron density than the packed acyl tails of phospholipids. Okay, so those were our signs of sterile, but what about PIP2, a lipid that's known to be important for isosome assembly and function? Well, first, the pill, like many bar domain proteins, has these patches of dense positive surface charge on the concave face of the banana where it would bind the membrane. And these, of course, would be ideal sites for binding a negatively charged PIP2 head group. And in fact, when we look in our EM maps, we can see some unassigned density sitting in this membrane-facing pocket. And that unassigned density is coordinated by all of these positively charged residues. I think it's a pretty good candidate for a PIP2 head group, but in these near native samples, I didn't really have the resolution to orient a PIP2 molecule or even say how many lipid head groups could fit in this density. So at this point, we could see these signatures in the membrane that could maybe be sterols and PIP2 head groups, but where to go from here? How do we test these hypotheses? So the way we did this was with in vitro reconstitutions and molecular dynamic simulations. We got the suggestion from Orléans Roux that we should try comparing our near native isosomes with ones that we reconstitute with lipids of known composition. So we started a collaboration with these guys from the Roux lab, Marco and Javi. We used purified PIL1 expressed in E. coli and reconstituted with lipid mixtures of known composition. We kept as a constant component a lipid mixture that's commonly used in vitro reconstitution experiments with bar proteins, which contains DOPC, a neutral lipid, DOPS, a negatively charged lipid, and DOPE, a conical lipid that's supposed to increase the tendency of the membranes to tubulate. Then on top of that constant component, we varied our lipids of interest, sterile and PIP2. We ultimately used 3 combinations, no PIP2, yes sterile, yes PIP2, no sterile, and PIP2 and sterile. So the first thing I wanted to look at in these structures was my region of interest near the amphipathic helix. So what you can see here is that the amphipathic helix density is totally missing in the samples that don't have PIP2, but in the samples that do have PIP2 but not sterile, we can see the amphipathic helix again. So what we can't know from these structures alone is whether the amphipathic helix is not folded at all in the absence of PIP2, or if it's just mobile, and that mobility causes it to be blurred in our structures. So Marco was able to answer this question using CV spectra on a synthetic peptide of our amphipathic helix. And those data showed that the presence of PIP2 allows our peptide to fold into a helix, suggesting that our missing helix here is actually not properly folded. Okay, so next I was interested in what was happening with this void pattern in the reconstituted samples. So here I'm comparing equivalent slices through the native-like membrane to a tubule with PIP2 but no sterol. And you can see that the void pattern is basically missing in these samples. But when we add in the sterols, incredibly, we see that void pattern, suggesting that the sterols are indeed responsible for these voids. To confirm these observations, we asked Josephine from the Vani lab at Uni Freiburg to run molecular dynamic simulations for us. So this is her result. This is a model of PIL1 in which the residues of the PIL1 dimer with the highest occupancy of the sterile head groups are highlighted. What became clear to us is that the highest sterile occupancy was centered around aromatic residues. And that's F32 here, Y40, F42, and F50. And incredibly, corresponding to the locations of those 4 residues in 3D space, we see these 4 voids. So we're able to see these signatures of sterol in the membrane, and they're coordinated by aromatic residues on the amphipathic helix. Okay, now let's move on to look at the lipid binding pocket in our reconstituted samples. So here's the lipid binding pocket from a tubule reconstituted with PIP2 but no sterol. You can see that this is a much clearer PIP2 head group density than the one in our native-like samples. It has the triangular shape of an inositol triphosphate group, and these positively charged residues are all interacting with the phosphate groups. But something's missing here. When we compare the lipid density to the one in our near-native samples, we can see that this PIP2 density only accounts for part of the density. So what's going on here? What is this extra density in the near-native samples? The answer to this question became obvious when we looked in our plus PIP2 plus sterile samples. We can see a clear PIP2 head group here in the same location. But between that PIP2 and the amphipathic helix, we saw this extra density in blue. This extra density actually comes from our constant component lipids. It's a phosphatidylserine molecule. You can see the head group here that I've labeled with a pound sign, and we are actually able to build these 2 acyl tails labeled with the little stars that have a big splay. But why does including sterols in our lipid mixture result in the binding of this PS lipid? Well, one idea is that the sterols could serve to fill a packing defect created by the binding of PS to PIL1, which could stabilize the conformation that we see here. Okay, so to summarize what we learned from the reconstitution experiments, we found that PIP2 is necessary for the proper folding of the amphipathic helix. We showed that the voids that we observed in our near-native isosomes are in fact individual sterile molecules localized near aromatic residues on the amphipathic helix. And we showed that our lipid binding pocket is capable of binding a PIP2 head group, and also in the presence of sterols, that charged binding pocket can bind both a PIP2 head group and a phosphatidylserine lipid. So for the last part of the story, which I think is the most exciting, I'm going to show you our data describing a mechanism of mechanosensing by the isosome. So for this, we're going to return to the near-native isosomes, knowing what we now know about the lipids sequestered by the isosome lattice. So in this video, what I did here was I used symmetry expansion and density subtraction to create a single particle data set from all of the individual PIL1 dimers from each of the 9 native-like helices. So I used all of that data to do a 3D variability analysis. And this shows us the the continuous 3D flexibility in that data set. So what we can see here is an this region in which the N-terminal contact sites between the dimers are, we see this sort of spring-like stretching and contraction. Now, this region is connected to the amphipathic helix, which is also moving in response to that stretching at the N-terminal contact sites. And this movement would, of course, affect the lipid binding pocket. And what we can see is that this movement is in fact correlated with a change in the size and shape of that lipid density. So to dive deeper into what's going on here, I extracted 10 particle subsets along this axis of variability from most compact to most stretched, and then independently refined 10 structures to high resolution. So we're looking at 5 of them here. I chose the slice in which we could see most clearly the void pattern so that I could see what was happening with the sterols. And what you can see is that in the most compact structure, the void pattern is very clear, but as you increase stretch, those voids begin to blur, suggesting that the sterol molecules are more mobile when the isosome lattice is stretched. Next, I looked in the lipid binding pocket. So here are the 2 extremes, the most compact and the most stretched. And in the most compact structure, I actually see a better resolution of the lipid density here, starting to be able to see the separation between the PIP2 and the other lipid densities, suggesting that there could be a phosphatidylserine lipid here in the native structures as well. And on the other hand, in the most stretched conformation, that lipid density is blurred. And in these sharpened maps, you basically don't see any density in that pocket at all. So this brings us to our model for how the isosomes function as a mechanosensitive organelle. Under steady state conditions, the lattice is in the compact state in which the charged lipids are bound in the lipid binding pocket and sterol molecules are coordinated by aromatic residues on the amphipathic helix. Upon stretch, the lipid binding is, upon stretch, the amphipathic helix is, oh my goodness, sorry. When the isosome lattice stretches, this pulls on the amphipathic helix, destabilizing the lipid binding pocket and freeing the bound lipids, presumably causing a change in the fluidity of the membrane beneath the isosome lattice. This could then free the isosome resident proteins to go enact their signaling functions, and perhaps these lipids are serving a signaling role themselves. So with that, I'd like to thank a lot of people, obviously Robbie and all of the people in his lab, but also my collaborators, Ambroise, the people in Orléans Roux's lab, Marco and Javi, and the people in Stefano's lab, Josephine and Pablo. Thank you, and I can take any questions.